Hello, and welcome to Beyond Biotech, the weekly podcast from Labiotech. I'm Dylan Kassane, and this is episode 171 for the podcast. Today, I'm joined by Hector Garcia Martin, a staff scientist at Lawrence Berkeley National Laboratory. A pioneer in metabolic engineering and computational biology, Hector has spent over a decade decoding microbial systems, everything from termite guts to genome-scale flux models, to unlock sustainable biomanufacturing. Today, he's leading the charge in self-driving labs, AI-powered robotic systems that automate experiments, predict biological behavior, and accelerate the design of microbes that produce fuels, medicines, and materials. In this episode, we'll dive into why biology has traditionally proven so hard to engineer, how AI and robotics are changing that, and where this revolution is headed next. I hope you enjoy my discussion with Hector Garcia Martin.
Hector, welcome to Beyond Biotech.
Thank you, Dylan. It's a pleasure to be here.
Hector, now you're an American citizen, you're living out in California, but you studied and you came from the Basque Country. What attracted you to move from that part of the world over to the US?
Yeah, that's a long story. Yeah, I was born in Bilbao, in the Basque Country, and when I was growing up, I had a lot of questions about science, about how the world was made, and one thing that I realized is that a lot of the things that we used at the time, the kind of computers and video games that I was really excited about and interested in, that none of that was made there, right? It was all done in the US, right? So I got started interested in science, and when I got interested in science, they also told me like, oh, you should go do a PhD in the US. That's where the business is really made, right? So little by little, through a series of steps, I got first to Champaign-Urbana in Illinois, and then ended up in the Bay Area here, which is arguably one of the main generators of innovation in the whole world, all the way from AI to semiconductors to biotechnology to science and everything else, right? So that what really attracted me, the ability to make things and to be there with the people who are actually making the future, were making the science and technology that was actually confirming our lives, right? So that's what really brought me here in the end. I do miss the food from the Basque Country dearly, but I go there, a couple of times a year to bring my granddaughter to my parents. So my daughter to my parents. So.
Hector, your website says that you're a physicist working in synthetic biology. How does that happen?
Yeah, it's a very non-traditional career path, right? And so this all started. I was very interested in math and physics when I was a kid, and what attracted me the most about is the ability to predict things, to be able to jot down a few mathematical formulas, make some computations, and be able to predict. This is going to jump this high, or this is going to fall at this speed, or predict things. So that really, or this magnetic field is going to change these object by this amount, right? It's going to move it by this amount. So all of those things was really fascinating. The ability to use your brain and mathematics to do that and predict that and control that, for me, that was absolutely fascinating. And the other thing that really fascinated me was biology. I got a lot of questions about biology very early on that no one really could answer, right? One of my big questions initially is like, why do we sleep, right? It doesn't really make sense that we spend 8 hours of our day sleeping, right? Because evolutionarily, it's like 8 hours where you're completely helpless to any predator, right? So why? Well, what is the reason that is 8 hours and not 2, or why does every mammal or every animal pretty much sleep, right? So I was very excited with these questions. I remember also when I was a kid, my dad told me that in the Permian era, there was these dragonflies that were like 75 centimeter big. They were like a third of us. It was just a dragonfly, right? And I was fascinated by that. And the reason is that the oxygen in those times was like around 30%. So it was much higher than now. And because the insects get their oxygen not through lungs like we do, but through tracheas and through diffusion, the higher concentration, the bigger they could be, right? So that concentration of oxygen determined that you could actually have much higher insects than we do now, right? So that was fascinating for me, and be able to predict those things and do those calculations was really fascinating to me. But at that time, biology was not like that. Biology was a lot of memorization and learning the parts of the body or the parts of the TSA cycle or things like that, which was utterly boring to me, right? So I was fascinated by the ability in physics to predict things and control things and use math to do that, but also by the biological objects, right? I was unable to decide what I wanted to do with my lives, and so I went through the path in which I had to defer that decision the most, which was physics, because I was told, like, look, if you do physics, you're trained as a problem solver, and you can do pretty much anything you want out there. And that turned out to be true. My physicist grad student peers, are doing anything from finance to synthetic biology to material science to anything in between, right? So I went into physics and to quantum physics. I learned a lot of very, very, very interesting things about emerging properties that I think will be critical for the development of biology in the future. And at some point, when I did my PhD, I started working with my advisor, and he definitely was a typical, well, he was not a typical physicist, but he had a very typical physics attitude that biology is just a lot of frozen accidents. So there is nothing really to learn there or predict. It's just things that happen, right? And I said, like, look, we study complexity, right? The complexity is the science that studies, how when you put several parts in a system, the behavior is much more complex than the sum of the parts. And it's like, and the best example of complexity are out of this window, and it's green, right? And we had a lot of discussions. Eventually, he changed his mind. He claims it wasn't because of me, but he did change his mind, and we work in biology. And we started, on one side, we did our physics project with Bose-Einstein condensates, which is some very esoteric physics systems. But we also worked on something called the species area rule in biology that tells you that the amount of species in an area grows with the area as a power law. So the number of species is the area to the 3/4. And that exponent, the 3/4, is always the same, right? It's pretty much the same for bacteria or birds or monkeys or fish, right? And that was really intriguing to me. And as part of my PhD was actually showing why that was the case, why it was universal and why it was a power law, right? And that was one of the papers that I wrote for my PhD. And then I did some other work on physics. And at the end of my PhD, it's like, well, Hector, you can go into more path-integral Monte Carlos and condensed matter physics and do what other people are doing, or you can just go into biology where there's definitely a need for making predictions, right? And I thought it'll be like, oh, a whole field for me, I'll be great. So I did that. I took, I suppose, like a kind of a biology position, doing data analysis in the Joint Genome Institute with the genomic analysis, and that was interesting. But a couple of things came up to me. It's like, one, that most biologists didn't give a damn about predicting things. They couldn't care less, and that was a bit of a downer. And the second thing is that a lot of the papers that we wrote, some of the first metagenomic analysis where metagenome is when you actually go into a microbial ecosystem and you take DNA pieces, you put them together to figure out all of the biochemistry of that microbial community. And we put some of the first papers out, for an enhanced biological phosphorus removal microbial community and a termite gut that was eating wood and converting that into feed for the termite. So we did a lot of interesting things, but I realized that all of that was descriptive. None of it was predictive. We gave a blue map, of where all the carbon and all the food was going, all the energy was going, but we didn't really do a lot of experiments to make predictions to actually test that in a quantitative fashion. And that was a bit frustrating to me. So when I met Jay Kisling, who was doing synthetic biology, I said, like, wow, this is what we need to do. We need to go into synthetic biology so we can make models, and then we can perturb it with the synthetic biology tools. And synthetic biology, what it does is it's a type of biology where it's trying to change the system that you're working with, right? In this case, the cell, you change the DNA to change the metabolic pathways that create this or that metabolite to produce more or less or change, the cell behavior to a specification, right? And I say, like, well, these people are doing just this. They are really changing the genome to make predictions. So they were going to need more people like me. So that's why I got feet first into that. And it was initially a complicated situation because I was a theoretician, right? I was a theoretical physicist. I was trying to theoretical physicist, had my papers, but I just did what is now called a data scientist. And in those times, it was kind of a bit, it was called bioinformatician in biology, right? Because the only people who did those things were the people who actually deal with the genome, with the genetic data, right? So it's kind of new. And then I went into a place where it was mainly, when I went into the Genome Biology Institute, that was funded and I helped write the proposal for. When we went there, it was mainly an experimental show where there was very little appreciation or understanding for theoretical aspects or whether biology could be predictable. Many people told me like biology cannot be predicted. It's like Jurassic Park. It never happened, right? It's like there is all these things that you cannot control. You cannot control biology, and it was it was a tough time for a while, but over the years, and it's been now 16, 17 years, we've created several predictive models that are able to predict synthetic biology and biology in general. And we are starting to create predictions that are close to what you can do in physics, to the kind of, or in chemistry, right? To the kind of predictions that I was doing in my PhD when I was looking at the vortex profiles of these post-synaptic condensers, which are like these very slowly interacting gases that you actually put at very low temperatures. So you would see the quantum properties. And, when you have a quantum liquid, and if you rotate it, you have a normal liquid, you have a big vortex. But if you have a quantum liquid, what you have is like a network of little vortices put it together. And that's really the hallmark of the quantum effects. And I was able to predict the whole vortex profile within 5 to 6%, right? It was very good predictions I could do. So we're now getting to the point that we can start making those kind of predictions in biology and synthetic biology. And it's been a lot of technical work of finding the tools and the experiments and the data quality that makes that happen, but also a lot of sociological work, and help people understand that it's not only this is possible, but this is desirable, right? And it's particularly useful in biomanufacturing because otherwise, the kind of changes you can do is just insane. It's exponentially big, and you can't just do all of that. So you need some kind of guidance to make that happen. And we had a few papers that are really show how you can apply these methods and get results that are really hard to do without applying predictive methods, right? So it's only lately that we have the appreciation that all of this really makes a difference in biology, right?
Hector, you're at Berkeley Labs now, and you've been there for a couple of years. For people who aren't familiar, can you just give me the short version? What is the Berkeley National Laboratory?
Yes, that is a good point. People know about universities, but they don't know what national labs are, typically. So national labs are large institutions that do what we call large-scale, team-oriented research, right? A lot of what happens in a university are like single PI groups where each grad student or postdoc have their own project and they just go with it and make it happen. But around the 1930s or so, it was starting to become obvious that at least for research in physics, that model was not sustainable, right? And there were some things that you couldn't reach in such a way. And that's why Ernest Lawrence, the creator of the Berkeley National Lab, came up with a different way of work in which you had a multidisciplinary team with scientists, engineers, and all kind of people contributing to a single thing. And that eventually led to all of the synchrotrons and the light sources that we use to get the structure of proteins, right? If you look at the advanced light source right now in Berkeley, it's not like a single PI person that has a synchrotron in their group. No, it's like many people put together that create a big facility that has many users from different places that can use that, because that infrastructure helps science. It cannot be done by a single group, right? So that's what the national labs really focus on, like these large-scale projects that are multidisciplinary that enable science at industrial scale beyond like the artisanal approach that has been the hallmark of science, right? And Berkeley National Lab is the first one, arguably, but I think it's the first one. And has been doing that for almost 100 years. I think it's 95 or so. I think, yeah.
Hector, if we can talk a little bit about synthetic biology a little bit more. You talked about prediction a minute ago. That's always been one of the really key hurdles, is our inability to predict biological systems. They don't act like physical systems. Can you sort of elaborate on why traditional methods for trying to predict biological systems have been slow, costly, and just inaccurate? What's wrong with them?
Well, the problem that we had in predicting biological systems is that the traditional approach in microbiology and synthetic biology has been very mechanistic, right? The idea of microbiology was like, well, if we understand the mechanism, we just need to understand all the mechanisms, what creates what affects that, and then we'll put all that together and we'll be able to predict, right? And that has failed for the last 70 years, right? And the reason for that is because, well, not everything is, we don't know all the mechanisms, and finding all of the mechanisms that that play a part is really hard because there's a lot of them, and we wouldn't know exactly which ones actually make a difference when we're looking at. Second of all, because there are many things that we don't know about in terms of not just the actual mechanism that it does it, but phenomena that we don't know about, right? Like emergent properties, right? One of the things that is done in condensed matter and in solid-state physics is that there's some systems, when you put together several small pieces of a system, it behaves in such a way that is not encoded in each of the parts, right? And that is something that we know happens in biology, but there is not a lot of documented examples of that, right? So, and that kind of emerging properties approach has been, or complexity approach is something that is not typically considered in microbiology, right? So it's partially that, right? And then what that has led is that, and the final thing is that there hasn't been a lot of interest in making quantitative predictions. And that's the only way to really know whether you are predicting things right? If you're making qualitative predictions like this will go up or down, there is a lot of times you can get it right for the wrong reasons, right? Whereas a really, truly rigorous approach will make a quantitative model, right? So I think, and that's had led us to people in synthetic biology just trying all possible things, right? When you are designing a pathway and you want to get the promoters that will tie the polymerase to express a protein, you just try the whole library and see what happens, right? And that is okay if we have like 5 genes because there's only, a combinations you can do is like 8,000. But if you want to do like a whole genome with 3,000 genes, that's unbearable, right?
So tell me, how does this unpredictability then, and it's really fundamental unpredictability a lot of the time, how does that complicate things when it comes to biomanufacturing?
Well, it does because you have to try a lot of things before you can find the cell that is producing your product at the desired tighter rates and yields and at large scale, right? So the way that it works right now is like, we are going to try all of these different parts. We're going to do 8,000, 10,000 experiments until we find something that produces tighter rates and yields, that are high, like let's say like 80 grams per liter, more than 90% the theoretical yield and so on. And then we're going to put it at a large scale. And then what happens typically is that things at large scale are not like the ones at lower scale. So you have to come back and make some experiments to try some more things, to try to figure out why it didn't work at scale up. And maybe, there is a toxic byproduct, or there is not enough oxygen, or there's some acetate, and so on, right? And there's like this back and forth between different scales. And the small scale experiments are rather inexpensive and easy to do, but the large scale experiments are really, really expensive to do, right? So to give you an example, and I always give a similar one, for Ameris to produce artemisinin, it took more than 150 person years, right? Where a person, if you take the number of people for number of years as the person years, so that like 150 years will be 15 people over 10 years, so a big time, right? But if someone from 2025 would have gone there to Ameris at 2005 or 2004 and told them, look, use this strain with this DNA code, put it under these conditions, and then scale it up under these conditions, it'll take them perhaps 7 to 9 months, right, to do it. So everything else from 9 months all the way to the 10 years or 15 years that actually took, all of that time is just searching in the phase space, right? It's like, let's change the promoter, let's try these conditions, let's try this media. If you could predict that, maybe if it wasn't perfectly, if you can have some prediction, instead of doing 1,000 experiments, you can do perhaps 2 or 3 or 10, right? And that is how being able to predict things can really accelerate biomanufacturing.
One of the things that's really interesting about the work that you're doing and what brought us to you today was when you start talking about the nexus between biomanufacturing, bioengineering, and artificial intelligence. And now you're really pioneering this use of AI and robotics in something called self-driving labs for synthetic biology. Before we dive too deep into it, can you start by telling me what is a self-driving lab and how does it speed up discovery? Why would I want one?
Yeah, well, that's a good point. So this all started by using, started using AI to predict things and tell bench biologists like, look, try this DNA sequence, try these conditions, and so on. And then you realize that often the bottleneck is our ability to do experiments, right? And so the next thing is like, well, let's couple this AI with a robot that does the experiments just as the AI that wants it, right? Because, humans, they have their own biases. They're like, I'll never do that experiment because that's never going to work, or I want to be on vacation, or I can only do so many things, per day and so on. A robot, can work 24 hours, can do a large amount of things, and it tells you, and it does things exactly as you tell them, right? Even if it's, if it's against the common sense of a human person, right? So that coupling of robots and AI, the idea is that it will make things incredibly much more effective. And the idea is not just to stop at robots and AI, but actually couple them together into and miniaturize them into microfluidic chips that combine, the AI and the robots into small chips that can be printed for like $800 and they can fit in your palm. And the idea is that if you can able to achieve that, then you can scale that up because you can print all those microfluidic chips, at $800 a chip, and expand that systematically, right? So Self-Driving Lab is really a device that combines the AI that generates, but that decides which experiments to do next, and a set of robotic robots that actually create produce those experiments, create the data, collect the data, and pass it to the AI in a loop, right? And the idea is that will accelerate science significantly.
Does the AI help overcome any of those challenges around prediction that you were talking about?
Oh yeah, that's the thing. What I think is very critical to understand is that many of these AI tools don't really need to understand mechanism, right? And we can actually make very good predictions without understanding all the mechanisms involved. And that perhaps is not so good for science, but it is great for biomanufacturing and engineering because it helps you design the strain that you want to get a particular specification, right?
So from this research that you're doing then in these genome scale models, the proteogenomics, how are you integrating AI to actually optimize enzyme expressions or pathway designs in microbes for better production of things like biofuels, of medicines, of polymers?
Well, I'll give you an example that we just are about to publish. It's going to come out very soon. In this case, where we were using, we had a Pseudomonas putida, which is a bacteria that was engineered to produce this a molecule isoprene, which is a 5-carbon sugar that you can combine into something called DMCO, dimethyl cyclooctane, and that is a sustainable aviation fuel. That's an aviation fuel that has an increased energy density. So if you use that DMCO to power your plane, you can get 8 to 9% further than you would with a normal fuel, right? Because it has a higher energy density. So in this case, we had a basal production of around 150 or so milligrams per liter of production of isoprene, and we said like, well, what we need to do is mess with the host metabolism to direct as much carbon in the direction of the pathway, right? But it's not clear how to do it, because there's like around 6,000 genes, or 6,000, 5,000 reactions, 6,000 reactions. And, if you downregulate one, then it may go in a different direction. It's really hard to predict which way the carbon is going to go and so on. And there's all really interlaced. So it's a complexity problem, an emergent complexity problem, right? So, and we have these tools now with all these CRISPR. We have these CRISPR-I tools, CRISPR interference, that allow us to take a gene and downregulate it instead of, producing like 10 proteins, it can produce one protein, right? But choosing which of them and which combination to increase production of your isoprene, that is highly non-trivial because, as I said, it's like, if you can do guides that downregulate 4 of these, and you have like 6,000 times each time, the amount of combinations is on the order of billions. So what we did is like, well, let's use AI to predict which which genes to downregulate to increase the production of isoprene. And that's what we did. We started with a few single guides. We collected the data, put it into our algorithm, that we call the automated recommendation tool, that recommended new combinations of genes to downregulate. And we did around 6 DDL design, build, test, learn cycles of that. And in 6 cycles, we increased production around 5,000%, from 150 to all the way to 800 milligrams per liter, right? So, and this is just a test of that. Now that we know that it works really, really well, we want to keep expanding that, right? So, and some of the combinations that it produced were like really unintuitive and would have never been possible to do through traditional methods by understanding the mechanism, because most of the genes in Pseudomonas are not annotated, right? We don't know what they do, so.
That's an incredible difference in scale there. I guess, who's the market for a self-driving lab? Who's going to be listening to our podcast today and thinking, I want one of those, I need one of those? Who's it aimed at?
Yeah, so I think there's 2 kind of users of self-driving lab. And, but for the record, the idea of self-driving labs, it didn't start with us. It became very popular in material science and chemistry first, and now it's becoming very popular in synthetic biology. And it's taking the world by storm. China is investing significantly in self-driving labs. There's a lot of interest now within the DOE. So it started to become like a really interesting idea to many people. And the people who are mostly interested are scientists, from the science side of things, where they can actually create a self-driving lab. And then they can, in principle, I think we could automate the creation of knowledge, right? We will be able to put a cell into our self-driving lab, as I was telling you, and it will create, a map of what the cell does and how it behaves when things are changed. And it could create what we call a digital twin of the cell that we could use to predict what would happen if we made genetic modifications or we changed the environment or things like that. So from a scientific point of view, there is the tantalizing possibility of automating creating scientific knowledge. And how you encapsulate that, whether that's going to be in an AI, in an LLM, a digital twin, or a set of papers that something writes, we don't know. But we could create scientific knowledge systematically. If you think, the variety of different organisms that it's out there, imagine that you create a self-driving lab for each of the 8 million organisms that may be out there. And within a few days, you have digital twins for all of them in a systematic fashion. So you can use that to do microbial engineering, to produce interesting things, produce new antibiotics. That could be of interest, right? So that's science. The second one is the people, industry who's doing biomanufacturing, and they don't necessarily care, why things happen, but they want to make things happen, right? And they have in this case, they have a Pseudomonas spt that produce a small amount of an aviation fuel, and they want to increase the titer rate and yields in a matter of weeks instead of years. They could be interested in that because it has some possibilities out there, right? So it has both practical applications and scientific applications.
I can imagine that there's top pharma companies, big pharma companies, who would be interested in something like this when it comes to scaling. But it also would help a smaller biotech really step up and play on a different level. Can you see both groups being interested?
I see both groups being interested, and I think it's the smaller startups that have the upper edge here, because it really requires changing the way you do things, right? And it's easier to be disruptive and change things dramatically in a startup. So, but of course, the other thing is that you need a significant amount of resources to do all of this automation, right? So we've seen big companies taking up this, but we've seen an a bigger uptake in smaller companies.
Let's talk a little bit about the future. You're someone who's been working in science, whether that's physics or synthetic biology, for a long time now. Where do you see the field evolving in the next 5 years, considering the advances we've had just in the last 5 years?
Well, we are in a very peculiar time in science as a whole, in which there's one technology that seems to going to have a disproportionate effect, which is artificial intelligence, right? And the reason for that is that AI is a bit like electricity. It can be applied everywhere, and it also because it really is the first technology that is able to do what humans do, right? So it's really hard to predict what's going to happen long term. First of all, because AI has a lot of possibilities and a lot of a lot of potential, but it's also true there's a lot of hype, right? And at what point, the full potential is going to be really embodied, or we just going to have a lot of failures? We don't know, right? But I think that AI is here to stay. It will change science the way we do it. To what point? I don't know if it'll change it dramatically to the point that in 5 years' time, when my kids start studying science, they'll talk to an AI instead of a teacher, or it'll be just another tool that is nice to have, but it has some important limitations. We don't know what that is. So AI is going to change that dramatically, right? And I think robotics will help with that and leave a resurgence in not just in biology, but all sciences. As I said, material sciences, chemistry, biomedical research, all of them are integrating these things. So that's how things are going to change. Now, how many self-driving labs we'll have or not, I don't know. There is some, the software side of things of AI works real fast, but once you get into the physical world, timelines are much slower. And once you have like an exponential curve from software and AI, much a linear curve, of productivity improvement from physical things, usually the linear trend wins. But we don't know what's going to happen there. So I think it will change science in the sense that it will eliminate a lot of the boring work and routine work. The things that you've done once, you will be able to tell an AI to just do, an agent to do, and so on. In terms of like really surprising things, I do hope that AI, is able to give us completely unexpected ideas because it has perhaps less biases, and perhaps it can integrate much larger amounts of data, and it doesn't have to be retrained every time, right? If you think about our lives, we are born. I mean, I have a 3-year-old right now, and I spend all my time explaining her things that we already know, right? So we are taught, we're teaching ourselves all of our lives, and then it comes to a point around, 30 or 40, where you actually get to the end of human knowledge, and then you make a little increase of human knowledge, you make your contribution, and then you retire, right? And then you start over again, right? So, and AI, doesn't have to go through that, right? And that's why, we expect, a lot from that. Of course, that such a disruptive change will have economic and social consequences as well, and that's a different conversation. But I think it'll change how we do science, and we need to get ready for that, right? And it's not everyone is ready, right? I'm not sure that I am even ready for that too.
Hector, what are the trends in AI, and particularly when it comes to biomanufacturing, that really get you excited?
Oh, I'm excited about the ability of these new systems to do reasoning and collect large amounts of data and give you a reasoning of why they make some type of predictions, and to do that in a more systematic way, combined with experimental work, right? Right now, we have like these reasoning systems that don't really, they tell you like, I'm gonna suggest this because of this, and that, but they never check that. If we're able to check that systematically with experiments, that you can run, through cloud systems or things like that, that is when scientific knowledge can really, really explode, right? So that excites me. I'm excited about that. I'm really interested in the whole idea of digital twins, because of my predictive interest, how to actually create quantitative models that are really able to predict things that we couldn't, and use that to its full extent in biomanufacturing, not just to create interesting, biomaterials or fuels or whatever, but also to create new therapeutic agents and also understand medicine much better, right? Can we create a digital twin of a human being? Can we understand, diets, the diet fats, I love to be able to have like a therapeutic theory that tells you, what is a good diet and why not, and why is not a good diet and why, right? That's something that we don't understand very well. We love to have digital twins that explain consciousness. What is consciousness? How does it create it? How does it get created? So all those things I'm very excited about. How will that happen? I'm just not sure, but we're going to do our best to contribute to that.
Hector, what should I be keeping my eyes out for when it comes to these self-driving labs and this new way of doing science? If I'm just living my everyday life, where am I likely to see an impact of these self-driving labs?
Yeah, well, that's a very, very, very good point. I think you will see you're going to see an impact in in therapeutics, in like new drugs being created systematically, instead of having like the list of FDA approved and the other, like which maybe, I don't know, 20,000, 30,000, 40,000, you're going to have the ability to create molecules to a specification. It's like you're going to like, I need a molecule that binds to this muscarinic acid receptor, but doesn't bind to this, other receptor, so it can alleviate, concussion here, and then you're going to be able to generate all of those molecules that will have those binding affinities, and then it'll give you the ability to create those systematically, right? So I think you will see that in therapeutics a lot. I'll see that. I think we will make biology predictable finally. I think you're going to see a bunch of new materials as well that may actually change things. So those are the things that I think is going to have an impact in to start with, right? So.
And finally, Hector, for aspiring scientists or for students who are listening who are really interested in this field, based on your own career path, you came to it from physics, you came to it with a curiosity. What sort of advice would you give them if they wanted to move into the field?
Yeah, that's thank you for this question. I really appreciate it, because I wish someone had given me this kind of advice when I was like 14, right? So the one thing I like to tell people when are starting this is that when I was 14 or 13, I didn't know what the heck to do, and I truly didn't know what the heck to do. And my instinct was like, well, let's figure out everything that is possible and then choose something. And that was, of course, impractical for a 14-year-old because no one explained it to you. But the one thing that I would say is like, look, the job that I have right now did not exist when I was 14. It just did not exist. I made it, right? When I was trying to make biology predictable, because I was really interested in why fireflies were 75 centimeters in the Permian area with 30% of oxygen and not 80 or centimeters or 2 meters, what stops a firefly from being 3 meters long? Is that even possible or not? I just followed my passion, right? And I think about it, how they do it with soccer players, where when they're looking for soccer players at age 4 or 5, they don't really look whether they are very technically capable. They look for some basic things, but the most important thing that they always tell to people at that age, is like, just don't correct them. Just make sure that they have passion for what they do and they love it. Because if you have passion for what you do, you'll keep doing it, and you will be able to overcome hurdles that no one else can really figure out until you're there, right? So I would say, like, look, there is a lot of things that don't look possible right now, but if you stick to them, your passion can make them important, and you can make it happen. It doesn't happen every single time, but you can always see what makes sense and what doesn't, right? So something that you're really passionate about and you can't stop thinking about, it's really you can morph that into your occupation and your day job, right? So that's what I would say, like, look out there for the things you're interested in, and the ones that you can't take out of your mind because you're always thinking about it, you can make a living out of that, particularly in this day and age. Right?
Hector, that's great advice, and thanks for joining us today on Beyond Biotech.
Thank you. It's been my pleasure. Thank you, Dylan.
That's it for this episode. Don't forget to check out all the latest news and analysis at Labiotech.eu. I hope wherever you are in the world, you have a great week ahead. Thanks for listening, and I hope you'll join us again next time for another Beyond Biotech.